All right, good afternoon, everyone. Thank you for joining us for today's webinar. I'm Cassie, Senior Marketing Supervisor at GenScript, and I'll be the moderator for the webinar today. So a little background about GenScript for those who are new to us. GenScript is the leading life science research and application service and products provider in various fields from basic life science research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. In this webinar today, we have Dr. Chengxun, our field application scientist, to present on cPass SARS-CoV-2 Neutralizing Antibody test for next generation vaccine development and immunity status monitoring. Dr. Su received a PhD training in biomedical engineering, interdisciplinary graduate school at Nanyang Technological University in Singapore. During her PhD, she focused on developing bioengineered platforms for disease modeling and drug testing applications, where she applied cutting-edge technologies in cell biology, microfabrication, microfluidics, and molecular biology to develop novel tools to support fundamental and translational research. Using these cutting-edge technologies, Chengxun spearheaded several collaborations with clinicians to investigate immune dysfunction and inflammatory profile in patients who recovered from COVID-19, paving the way for potential intervention strategies. This webinar will take around 45 minutes and we will have 15 minutes for Q&A session. You can type in your questions in the chat box and we will answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we will email the answers back to you after this webinar. Do also help us to fill out the short survey that you can see on your screen to stand a chance to win a GenScript gift pack. So now, without further ado, we will let Dr. Su to start her presentation now. Dr. Su, over to you now.
Hello, everyone. Thanks, Cassie, for the introduction. So very good afternoon to everyone, and thank you very much for tuning in for today's webinar. So before we start the webinar, I would like to just to know a little bit about your background. So if you could help us to type in your answer for the poll question. Are you currently working on SARS-CoV-2 serology or immune response related projects? Okay, you will have about 10 more seconds to type in your answer. Okay. So let me see your response. Okay, so now I see the response from you. So about 60% of you are currently working on SARS-CoV-2 serology related projects, and about 40% are not working on such projects. So that's good. So I think majority of you are probably already are using cPass for your projects. So today I will be introducing some of the most recent literature that will hopefully benefit your work. So this is the agenda for today's webinar. So first of all, as usual, I will give an introduction on what is neutralizing antibody and the technology behind cPass. So for those of you who are already a cPass user, I apologize in advance for you to have to repeatedly hear this, but I promise I will make this brief and useful. So, and then secondly, I will introduce the clinical performance of the cPass test kit, as well as the results expression and interpretation. So thirdly, I will introduce how can we leverage the SVNT platform for detecting variant-specific neutralizing antibody. So in this section, I will be covering the most recent work related to how people have been using different platforms, including SVNT, to detect VOC-specific neutralizing antibody. And the last topic I'm going to cover is related to how long can vaccine last, and specifically how long can neutralizing antibody last after infection or vaccination, and how we can leverage cPass test as a tool for monitor whether we will need a booster or not. Okay, So first of all, just a little bit background information on neutralizing antibody. So our human body is designed in a way that once we were exposed to foreign particles, our immune system will produce antibodies. Whether the foreign particle is from an infection or from vaccination. So this is to protect us from the next exposure to the same antigen. So when the next time the virus invade our body, those antibodies that was produced before will act as the frontline soldiers to protect us from reinfection. So according to the function of the antibody, it can be categorized into the binding antibody and neutralizing antibody. So first, binding antibodies are those who can bind the virus, but it may not interfere with its infectivity. And on the contrary, a neutralizing antibody binds to the virus and interferes with the virus infectivity by directly blocking the virus from entering the cells. So this process is very well illustrated in this schematic over here. So you can see that for the SARS-CoV-2 virus, when it comes into contact with the cells, the RBD region on the spike protein will bind to the ACE2 receptor on the human cells, which will then trigger the internalization of the virus. However, if we have neutralizing antibody present in our system, this neutralizing antibody will bind the RBD region on the spike protein and in turn inhibit its interaction with the ACE2 receptor, which will eventually block the virus from entering the cells. So given this function of the neutralizing antibody, one of the key objectives of vaccination is to generate neutralizing antibody so that when we are exposed to the virus after the vaccination, we have those antibodies to protect us from reinfection. And also because of this functional aspect of the neutralizing antibody, it is frequently used as an aid to identify your level of adaptive immunity. And also a neutralizing antibody can be used as a therapeutics for treatment of COVID-19. So earlier on in the pandemic, those who are recovered from the COVID infection, if they have enough neutralizing antibody in their blood, their plasma can actually be used as a treatment for COVID-19, which we call them covalescent plasma, to transfuse those plasma into sick people so that the neutralizing antibody present in the plasma can help to fight the virus in the sick people. So next, I would like to introduce two very important publications related to how neutralizing antibody is correlated with real-world vaccine efficacy. So the first study I would like to introduce is the one published in Nature Medicine last year. So in this publication, the authors analyzed the relationship of the neutralization level, meaning the level of the neutralizing antibody with the protective efficacy of all the 7 vaccines over here. So the vaccine they analyzed include those that is based on the mRNA technology, Moderna and Pfizer, and also protein subunit Novavax, and adenoviral vector AstraZeneca, and as well as inactivated vaccine. So when we are looking at the results over here, we can see that there is a positive correlation between the neutralizing antibody level and the protective efficacy, meaning that the higher the neutralizing antibody the vaccine can induce, the higher the protective efficacy was observed in real world. So when we take a closer look, we know that mRNA-based vaccines are the strongest in the protective efficacy, and then followed by the adenovirus vector-based, and then lastly the inactivated vaccine. So the next study I would like to briefly introduce is the one that from Moderna phase 3 efficacy trial. So this was published in Science last year. So in this study, it is also revealed that the vaccine efficacy is positively correlated with the neutralizing antibody level. in this study, it was found that the vaccine efficacy is estimated to be 45 to 60% for vaccine recipients who are seronegative, meaning that they do not produce any antibody after they receive the vaccine. So for those group of people, is about one out of 2 chance their vaccine doesn't really work. And then for those who have the highest level of the neutralizing antibody, the vaccine efficacy can increase more than 98% for them. So here I would just like to read this statement from this publication. So it is stated that the current results support that neutralization titer is a potential surrogate marker, meaning biomarker, for Moderna vaccination against COVID that can be considered as a primary endpoint for basing certain provisional approval decisions. So indeed, several of the vaccine platforms, their booster dose regulatory approval is indeed based on the neutralizing antibody level. So here is the FDA fact sheet from the Pfizer and Chengxun vaccine. So you can see the highlighted region that the effectiveness of the booster dose were based on the assessment of neutralizing antibody titer. And then for the inactivated vaccine, they also use the neutralizing antibody measurement to determine the effectiveness of the third booster dose. So the same goes for the Novavax vaccine, where they also made an announcement that their vaccine booster can have 4-fold increase in the neutralizing antibody level, demonstrating its efficacy. So how do we measure neutralizing antibody? Conventionally, a neutralizing antibody is measured using the virus neutralization test, which involve the use of live virus and cells. As illustrated in this schematic, when there is a neutralizing antibody present, it will prevent the virus from infect the cells. Therefore, by quantifying the number of infected cells, we will be able to know how much neutralizing antibody is present in the sample. So for this method, because of the use of the live virus, it requires strict safety condition, which is the biosafety level 3. So this is an image of the kind of personal protective equipment you will need to wear if you work in such a safety condition. So this can be quite logistically demanding. And also the process of this experiment usually takes 3 to 5 days to complete. And because of the highly complicated experimental protocol, it requires highly skilled operator. And also this assay is quite expensive and not scalable. So the new methods which we introduce here, which is the technology behind cPass, is called the surrogate virus neutralization test. So for this, in this method, we use the purified protein, which is the RBD, and also the ACE2 receptor to mimic the virus host cell interaction in a ELISA plate. So the RBD used in this method is conjugated with the HRP, so that when there's no neutralizing antibody present, all the RBD will bind to the ACE2 receptor coated on the plate, and which will result in a very strong signal. And in contrast, when there is a lot of neutralizing antibody present, those antibody will block the binding of the RBD to the ACE2 receptor. Therefore, this complex will be washed away, and there won't be a colored signal. So the advantage of this SVNT is that it only takes about 2 hours to complete the assay, and is automatable if you have any ELISA system that is currently in your lab. You can easily implement this technology to run with the ELISA system. And it also works in any laboratory setting as long as you have the regular BSL tool. And it can be done at a high throughput. So one kit contains 96 tests, and you can easily perform multiple plays at the same time. And also because of the standardized protocol we use, the results generated by different operators and also even different regions have observed to be very consistent. So next, I will review some of the most recent clinical performance for the cPass SVNT. So the first clinical agreement study we perform is using the PRNT50 as a comparator test. PRNT50 is the plate reduction neutralization test, which is the cell-based neutralization test that I introduced earlier on using live virus. So using this as the comparator, we have demonstrated the positive and negative percent agreement to be 100% in a cohort of 26 positive sample and 88 negative sample. So recently we have completed our another clinical agreement study using the PVNT50 as the comparator method. So PVNT50 is very similar to the live virus-based neutralization assay, except that it uses a pseudovirus, so the assay can be conducted in a BICL2 setting. And so for this study, we have accrued a much larger cohort with positive samples 313 and negative samples over 1,000. So as you can see from the result here, we have also demonstrated excellent positive percent and negative percent agreement with both more than 95%. So how about on a quantitative level? What is the correlation between cPass and the gold standard PRNT? So in this result shown here, we can see that the correlation of the results generated from the cPass SVNT have a very high correlation with that generated from PRNT, with an R-square of 0.9446, which is considered very high given any biological system. For those assays that is designed to detect the binding antibody, their correlation with the virus neutralization test have proven to be very poor, with R-square value less than 0.5. So the SVNT technology is commercialized under the trade name of cPass, standing for COVID Pass. And currently, we have obtained approval of in vitro diagnostic use in more than 40 countries worldwide. And I would just like to tell a bit of the history about the commercialization of this test kit. So after the ideation of this test kit, within 70 days, we managed to get the authorization from the Singapore HSA in May 2020. And right after 6 months, we received FDA EUA authorization. And we are also the first FDA authorized test kit for neutralizing antibody detection kit. So for the results expression, and we have 3 different ways to express the cPass results. The first and most straightforward way is to express the result in percentage inhibition by calculating according to this formula using the optical density you obtain from the plate reader. So for the percentage inhibition, the cutoff is 30%. With any value more than 30%, it means neutralizing antibody is detected. The second method to express the result is in unit per ml, which is also known as arbitrary unit per ml, using the neutralizing antibody standard, which is also available from us. So using the standard to prepare a calibrator of different concentrations, you can obtain the standard curve by analyzing using the 4PL model. And for this expression method, we have found that if the result is less than 100 units per ml, is considered low titer. This is equivalent for cPass percentage inhibition, 68%. So this low titer and high titer is based on the studies performed on investigational COVID convalescent plasma, meaning that for the titer that is more than 100 units per ml, or in other words, more than 68%, it is acceptable to be used as a convalescent plasma, and which of course is considered high titer. So a third method to express the result is in IU per ml, which is international units per ml. So for this method, we have this publication from the cPass inventors lab, who have established the mathematical model for converting the cPass percentage inhibition to international units per ml. So all you need to do is to download the Excel template from this publication, input the cPass percentage inhibition value to get the equivalence of the IU per ml. So I would also like to cover a little bit about the cPass protection threshold, meaning some of the studies that have actually investigated what is the level of neutralizing antibody that can actually confer protection. So the first study is from Israel, which is published in New England Journal of Medicine. So in this publication, during which the VOC circulating is the alpha variant, it was found that in a group of healthcare workers, for those who are protected, their cPass value is about 78%, where for those who have breakthrough infection, their cPass is around 60%. And then a second study is from Vietnam, during which the VOC circulating is the Delta variant. So in this study, there are two cohorts. The first cohort contains about 120 participants, and the second cohort contains about 40 participants. So if we look at a cohort with the higher number of participants, the results from this cohort is that among those healthcare workers with a cPass value of a little more than 80%, they are protected from breakthrough infection by Delta. So next, I would like to cover how we can leverage this platform for detecting variant-specific neutralizing antibody. So first of all, I would like to give a little bit background on what is the most current circulating variant. So this is the VOCs earlier this year in January. So we can see that in South Africa, in January, the Omicron BA.1 has started to displace the Delta, also the same for the USA in this period. So after 7 months until this month, we see that the trend of the evolution is that all the sub lineage from the Omicron start to emerge and displace the previous sub lineage of the earlier sub lineages. So for the South Africa, the majority of the VOC of the variants circulating is the BA.4 and BA.5, which are also first documented in South Africa. Where for USA, the main circulating subvariant is the BA.5 and also BA.2.12.1. This BA.2.12.1 is also first documented in US. So of course, we also know that the most recent, the newest variant is the BA.2.75, which is first documented in India. So what we know about Omicron is that it have a large mutation size on its spike protein with more than 30 changes to the amino acid sequence, where 15 of these changes is in the receptor binding domain, which is the most critical region that interacts with the human body. So the WHO have identified 3 key research priorities for investigating the impact of Omicron. So the first research priority is to identify whether the post-vaccination or convalescent plasma are still able to neutralize the Omicron. And then the second priority is to investigate the real-world vaccine protection of all the current commercial vaccines, which are still based on the Wuhan variant. And lastly, is about development of new treatment modality for treating severe diseases. So many of you have been asking whether we can use the SVNT for detecting neutralizing antibody that is specific to the variant. So we can very simply do so by replacing the wild-type RBD in the kit with a VOC RBD. So what you need to do is just to get an additional vial of the VOC RBD from us, and then use it as a replacement of the wild-type RBD in the kit. Then you can investigate the VOC-specific neutralizing antibody using the same protocol. So currently, the VOC RBD are for research use only, and in some countries where the regulation allows, it can also be used as a lab-developed test in diagnostic labs. So currently, the VOC RBD we have available include all the main VOCs, and we also have the newer variant, including the BA2 and BA4 and 5 available. So for BA4 and 5, they have the identical sequence of the RBD. So you just need to get one which can represent both. So here I would just like to point out our speed in developing such RBD protein for the newer variant. After the identification of the variant, usually we'll make the VOC RBD available within weeks after its discovery. So this is mainly due to the fact that we have the very strong expertise in gene and protein production, which enables the fast turnaround time for the newer variants. So here I would just like to add that riding on the wave of the growing biotech ecosystem in Singapore, we have recently established our gene and protein facility in Singapore with a local manufacture capability. So coming back to how to use the SVNT for measuring the neutralizing antibody response to VOC, the first study I would like to introduce is the one conducted in Thailand last year. So in this study, they investigated the vaccine efficacy for 3 different groups. So the first 2 groups received 2 doses of the Sinovac, and the second group received 2 doses of the AstraZeneca vaccine, where the last group, they received, the first 2 doses received is the Sinovac, followed by a booster dose of the AstraZeneca. So in that study, they investigated the neutralizing antibody response to the wild type alpha, beta, and delta. So we can see that when we compare the results between the 2 dose Sinovac and 2 dose AstraZeneca, we see that overall the AstraZeneca group have a higher neutralizing antibody level, and both of these 2 groups have the lowest response for beta, and slightly lower for alpha and delta. And when we see that when they combine the two vaccine with using a combined booster dose strategy, the neutralizing antibody response to all the 4 different variants have increased significantly. The second study I would like to introduce is a study conducted in Brazil. So in this study, we investigated 2 groups. The first group is those who are infected with the Delta variant. So when we test them using the Delta RBD and wild type RBD, we observe that they have a higher neutralization against the Delta RBD, which is expected because usually you will always have the strongest neutralization against the variant you are infected with. So the same goes for the wild type infected group. they have higher response for the wild type and slightly lower for the Delta. So another study is from US, which they investigated the response to the Delta RBD and Omicron RBD for all these different groups. So we can see that a general trend is that if you have high for people who have a high neutralization against the wild type, usually their neutralization against the Delta will decrease a little bit. Well, the response for Omicron will decrease even much lower. And we see that this trend is persistent for those who have received 1-dose vaccine, 2-dose vaccine, and also 3-dose vaccine. And we also see that those who have hybrid immunity, meaning that you have received vaccine and you also have infected naturally, those groups usually have the highest level of the neutralization. But some of them are still do not have a high neutralization against the Omicron. So just to note that for this group of cohorts, the variant they are infected with are not Omicron. So the next study is from Singapore, which is just recently published. In this study, they investigated the neutralizing antibody response for 4 different vaccine groups. So let's look at this column by column. So the first column is those who receive a 2-dose mRNA vaccine plus a booster dose of the mRNA vaccine. So when we look at their pre and post comparison of their neutralizing antibody response to the ancestral strain, which is the wild type strain, we see that before the booster, they actually have a huge variability in their capacity to neutralize the wild type RBD. Where after the booster, all of them have very high neutralizing antibody. However, when tested the same cohort with the Omicron RBD, we observed that for those, of course, before receiving the booster dose, we see the responses to the Omicron is very low. While after receiving the booster dose, most of them restore a high neutralizing antibody against Omicron. Well, some of them still do not have a good response. So this again highlighted that different individuals react quite differently to even the same vaccine. So the second column is those who received 2 doses of inactivated vaccine plus a booster dose of inactivated vaccine. So in this group, we see that the interesting result is with the Omicron here. So we see that even after receiving the booster dose, most of them still do not develop a strong neutralizing antibody level against the Omicron. And the third group is a combine of different vaccination. So they receive 2 doses of the mRNA plus 1 dose of the inactivated vaccine as the booster. So in this group, we see that for the ancestral strain, they have an increase after taking the booster. However, for Omicron, the response we see is still suboptimal. And how about those who is the reverse, meaning that they have received 2 doses of the inactivated vaccine as the primary series, followed by mRNA vaccine as the booster. So we see that when tested the response against the wild type strain, we see that before the booster, most of them their neutralizing antibody cPass percentage inhibition is below 70%, and after the booster, all of them have actually restored to a high level. When it comes to the Omicron, the results looks very similar to those who have received 3 doses of the mRNA vaccine. So from all this literature, we can summarize that usually for the neutralizing antibody response, when it comes to the Omicron, the percentage inhibition will reduce. So this is actually because I guess all of you would have expected because of the large mutation size on the Omicron RBD. So this is also termed as an immune escape of the Omicron. So in line with that, the neutralizing antibody standard from us, which is some of you may use it for developing the standard curve using the cPass. So we have tested this neutralizing antibody standard against all the different variants, including the newest subvariants. And for this standard, it is unable to neutralize the Omicron RBD, including all its sub-lineages. And we have developed a new Omicron neutralizing antibody standard, which is able to neutralize all the Omicron RBD and its subvariants. So when you do the semi-quantitative analysis with the Omicron RBD, you should use the Omicron neutralizing antibody standard. So because of its ability to neutralize the Omicron RBD, this antibody standard can also be used as the alternative positive control when you run the cPass assay. So how about the newer variant Omicron BA4 and 5? So we know that for when BA2 is identified, we know that its difference between the BA2 and BA1 is actually quite minimal. However, this is not the same case for the BA4 and BA5. So this is a very recent study that have demonstrated this point. So in this study, they investigated vaccine participants before and after booster dose. So this is a cohort containing 27 participants, and their neutralizing antibody against all the different variants were tested before and after booster dose with Pfizer with a 6-month interval. And all of these participants do not have infection history. So first of all, let's look at the before booster dose response. So we see that before receiving the booster, the neutralizing antibody level against the wild type have dropped to a relatively low level, where the neutralization against all the Omicron subvariants is essentially below the limit of detection, meaning a negative response. And after receiving the booster, we see that the neutralization antibody against all the different variants have increased significantly. But a very interesting observation is that we see the response against the BA4 and 5 is actually much lower than that of the BA1 and BA2. So this highlights that although the mutation size is only additional 2 to 3 mutation size, it actually really affects its ability to escape the immunity. So the second group is participants who are infected with the BA1 and BA2 subvariants. So in this cohort, those participants are infected with these two variants about 1 month ago, and majority of those participants have been vaccinated previously. So when we look at the results, we see that those who are infected with the BA1 and BA2, they still have the strongest immune response against the wild type. But of course, this could be due to that they are previously vaccinated, which the sequence is based on the wild type sequence. So when we look at their response to the BA4-5, we also observe that the neutralizing antibody against this newer BA4-5 is also much lower than that of the BA1 and 2. So in other words, even if people are infected with the BA1 or BA2, they may still get infected with the newer variant because it still can escape the immunity to a certain level. So just to give a summary of all the literature and recent studies about the neutralization to the variant of to the different variants. So basically, we know that there's a loss of neutralization observed, meaning that if you have a high level against the wild type, this level will decrease when it comes to the different variants. And this publication, which I'm going to introduce here, is published in Lancet last year. So it is a very good summary of all the existing studies in this field that have actually measured the neutralization of the variants compared to the ancestral virus. So in this table, you can see that when it comes to the VOC alpha, 1.6 times higher neutralizing antibody is required to achieve the same level of neutralization. While for beta, this fold change is 8.8. For the gamma and delta is a little bit over threefold. So this actually highlights that highlights the point that the more the mutation of this variant, the higher the level of neutralizing antibody you need to achieve the same amount of the neutralization. Of course, given that the neutralizing antibody is generated after the vaccination. So here, this schematic basically is a schematic illustration of this table right here. So meaning that for alpha, you need this amount. If it's beta, you will need a much higher amount, which is about 8-fold more. And the same goes for gamma and delta. So this actually gave insights to the vaccine makers on what should the vaccine development strategy be, given that the current variant circulating is the Omicron, and the previous vaccine developed is not able to generate enough immunity to defend us from the Omicron infection. So here I would like to show two pieces of news. So the first news is from Pfizer, who have recently completed an application for authorization of their Omicron-designed COVID booster. And we also heard the same from Moderna. And this news release is from Sanofi GSK. So for them, they took a different approach. So what they do with their VOC vaccine is they use a beta-containing vaccine, which they have demonstrated a relatively good efficacy against the Omicron. So with all the vaccine makers starting to develop the variant-specific vaccine, the question remains that whether the speed of the vaccine development can keep up with the emergence of the new variants, because we see that I believe all these Omicron specific vaccine are based on the BA.1 variant. However, the circulating Omicron subvariant right now is majority of them is the BA.4 and 5, so which the sequence is different from the initial BA.1. So it poses a very challenging question for the vaccine makers on develop the right vaccine, which can keep up with the speed of the mutation. So one of the strategy to overcome this challenge is to develop a variant-proof vaccine instead of the variant-specific vaccine, meaning that the vaccine is effective regardless of the different variants. So this sounds like a daydream, but it's actually achievable. So in a publication by the cPass inventor, Prof. Wang, so they have investigated the neutralizing antibody response not only against the SARS-CoV-2 and its variants, and also the SARS-CoV-1 family, which is the virus that caused the SARS in 2003. So in this panel of the virus, 5 of them are actually pre-emergent subecoviruses, meaning that they are now in the animal and are not yet circulated in human. So this study In this study, they have investigated 8 individuals who have recovered from SARS-CoV-1 in 2003, and they received one or two doses of the Pfizer vaccine. So very surprisingly, they found that for those who are infected with SARS and then followed by the COVID vaccine, they have developed a very high level and broad spectrum of the neutralizing antibody response. So this actually is very meaningful for vaccine developers to even consider a reverse way of doing this, so that to generate a very broad spectrum of neutralizing antibody. So last but not least, I would like to quickly cover how the cPass test can benefit the population at large, meaning its value in the public health. So the question we always ask is, how long do vaccines last? So we see from this graph, which is simply showing that we need a booster which causes a much greater response than originally, with much higher level of antibody and also higher affinity. So we also see that the antibody level wane over time. So with even for different disease, there are vaccines that can last for a lifetime after we complete the full regimen, and there are also vaccines that only last for a few months or a few years. So what we can leverage using cPass is to monitor the neutralizing antibody response at a very large scale because of its conveniency and it can be used in a high throughput manner and in an automated ELISA system. So you can see that in this study in Korea and also the study in Brunei, thousands of samples has been tested using cPass. So this study is also from the cPass inventor profile, demonstrating that for different group, their neutralizing antibody response can vary a lot. So I think I won't go too detailed in this because I believe most of you have already seen this publication. So another publication is investigate the persistence of the neutralizing antibody in New Zealand after 11 months recovery from the infection. So this study, the exposure is in March to April 2020, which is pre-Delta, and we see that even after 11 months of the recovery, they still have neutralizing antibody present in them. And of course, we see this high variability of different samples. And when it comes to the different variants, we also see that the beta has the lowest level, followed by alpha and delta. And of course, they also have the newest data on the Omicron, which we expected that all of the response to Omicron will be very low. So this study is investigates the neutralizing antibody response from inactivated and mRNA vaccine conducted in Hong Kong and Singapore, which basically tells the same trend. And what I would like to highlight from this study is that even though they are conducted in different regions, the results generated are very highly similar to each other. So this highlighted the high concordant cPass results from different regions. So all of us are interested to know whether a fourth booster is still needed, and whether by taking a fourth booster, it can offer some protection against Omicron. So in January earlier this year, Israel have administered the fourth booster for its citizens more than 60 years old. And so in this study is a summary of the outcome of this fourth dose efficacy. So we see that the blue dot is basically the efficacy of protection against infection, while the red dot is the protection against severe disease. So in this article, it was found that the protection against infection is very short-lived, only lasts for a few weeks. As you see, it quickly went down. And then we see the protection against severe disease can last for a much longer time. So in other words, it is still meaningful to administer the fourth dose, mainly to protect against severe disease, but not against infection. So what we would like to propose is that cPass can actually use forget and prioritize booster dose for those who have lower neutralizing antibody. For instance, within a population, there could be people with a higher neutralizing antibody and those with lower neutralizing antibody. So for those who have a negative or very low level, they are actually at higher risk of severe disease. So for those people, they can go for a screen in the clinic and decide they may go for the booster as early as possible. So another idea for the employers out there, you can also implement cPass as a corporate testing, as like a welfare to your employees to allow them to make a more informed decision on whether to take a fourth booster. So here I would like just to show our newest product, which is the neutralizing antibody lateral flow assay. It's a self-test version of the cPass test. So this, the image shows here, when you see the band, it actually represents the level of neutralizing antibody present by just using the finger prick blood. So the next, so I would also like to cover a little bit about monkeypox. So we know that WHO declared the monkeypox to be a public health emergency last month. So we know that monkeypox is actually not a new virus. It has been, it was reported in the 19th century in several countries, which have caused an outbreak before. However, earlier this year, in more countries worldwide, it has been reported again that this virus is causing another outbreak. So this is the monkeypox case trend reported to CDC. So we do see an increasing trend of the cases in US. And WHO have recommended that PCR right now is the preferred method of detection. So with that, we have also come up with a real-time PCR kit for monkeypox virus. And you can look into that if you are doing projects in this area. So in conclusion, so we know that the cPass SVNT platform permits the detection of a true functional neutralizing antibody that blocks the virus entry, and only a fraction of the antibodies produced post-infection or vaccination are neutralizing antibody. So with a lot of real world protection data and also scientific publications, neutralizing antibody has demonstrated to be a good biomarker for predicting protective immunity. And we also know that the antibody level wanes over time, and the waning speed is highly variable between different individuals, depending on what type of vaccine you take and your personal differences. And we also introduced that the cPass SVNT platform can be adapted to measure neutralizing antibody against the newer variants at large scale. And last but not least, we observe very highly concordant cPass results across different lab and cohort. So thank you for your attention, and I will take any question you might have or any comments or thoughts on this platform.
All right, thank you, Dr. Su, for the great presentation. We do have quite a number of questions coming in, so we'll go through it one by one. The first question, for calculation of percentage of inhibition, have you ever got minus values such as negative 20% inhibition? Why does it occur, and how do you interpret the results?
Okay, so thank you for this question. So yes, we do have seen such negative percentage inhibition result before. So this is mainly due to the difference of the matrix, meaning that the samples you test are the serum or plasma from different individual, and sometimes these samples can have a different background when you rate the optical density. So some of the optical density can be slightly bigger than the negative control. So this will result in a negative percentage inhibition value. So when you see such results, it actually just means that there's no neutralizing antibody. So so you can just interpret simply that is a sample that does not contain any neutralizing antibody. Yeah.
All right, thank you, Dr. Su. The next question, do you inactivate the serum before dilute?
For the serum inactivation, it is optional, meaning that you can either use it directly, or if you prefer to inactivate before you use, it is also okay. But you just remember that for the same project throughout, if you have inactivated in the first experiment, you just inactivate for the rest of all the experiments throughout the project to ensure consistency.
Thanks, Dr. Su. The next question, do you use negative control or PBS to normalize, to calculate the percentage of inhibition?
So for the negative control, we do provide a vial of the negative control in the kit for you to use to normalize all the results to the negative control. So basically, this vial of the negative control in the kit is the component, the constituents are just the buffer. So it's very similar to PBS. So yeah, it's just something you use to have a baseline level of the optical density so that you can normalize your result.
Thanks, Dr. Su. The next question, how does the neutralizing antibody response last post-vaccination versus natural infection?
Yeah, so this is a very good question. So the answer is that it really varies between different people. So usually the general trend, we see that if you are vaccinated against mRNA vaccine, 2 dose or 3 dose, likely your neutralizing antibody is going to last for quite a long time. Even after 6 months, you can still have a high level. And if it's inactivated vaccine, usually is lower. And also for the infection, so just now a very good study I showed is the study performed in the New Zealand. So you can see that even after 11 months of the infection, there still can be neutralizing antibody in some of the people. Yeah, in most of the people actually.
Thank you, Dr. Su. The next question, are there any studies on neutralizing antibody levels of breakthrough infections against mRNA or vector-based or inactivated virus vaccine across the globe?
So this again is also a very good question. So I would like to direct you towards a summary that I just mentioned. So this study actually investigates the neutralizing antibody level of the breakthrough infection. So in this Israel group, they are vaccinated using the Pfizer vaccine. Where for the Vietnam study, I believe it could be a different vaccine. So you can look into the article to see what is the vaccine they took of this population. So yeah, there are such studies, and each study, the cohort can have different vaccination. But right now, there's no such study that studied the breakthrough infection against Omicron. So this could be an interesting area that people can look into.
Thanks, Dr. Su. The next question, does the new Omicron-specific Moderna vaccine confers better neutralizing antibody response versus currently available non-Omicron specific vaccines?
Sorry, can you repeat the question?
Yeah, sure. So does the Omicron-specific Moderna vaccine confers a better neutralizing antibody response versus the currently available non-Omicron specific vaccines?
Yeah, so this again is a super good question. So the answer is that we don't see that data yet. So because these vaccines are still under the approval by the regulatory authority, and we I don't think they have released their data on this yet. But I think a very good publication we can look into is the one just now I introduced with this one. So you see that even for people who are infected with BA1 or BA2, so it can be considered that it will be very similar for the case they are vaccinated with BA1 or BA2. So for such people, they do generate a relatively high neutralizing antibody against these two variants, but it is slightly lower for the newer variant. So yeah,
Thanks,
we
Dr.
can wait for the data. Yeah, we wait for them to release the data, and hopefully the vaccine's effect is better than the natural infection.
All right, thanks, Dr. Su. The next question, what is the concentration range for A02161 recommended for standard curve preparation?
Okay, so this is related to the Omicron neutralizing antibody standard. So for this newer neutralizing antibody standard, the highest, if you are testing against the BA1 or BA2, you can use 8,000 unit per ml as a starting point. But if you are testing against BA4 or 5, we know that BA4 and 5 is a little bit more difficult to neutralize. So you can start with even higher than 8,000 unit per ml, probably something in the range of 10,000 or so. So that's something I would recommend.
Thanks a lot, Dr. Su. So we are running out of time. Take on the last question from the floor. The rest of the questions, maybe we will answer them via email. So the last question for today, do we know how many fold of increase in neutralizing antibody needed for the currently available vaccines versus Omicron subvariants?
Yeah, so this is again a great question. So I think this is related to this publication that I just showed on the different fold required for different VOC. So right now, there's no available data about this for Omicron yet. So yeah, I look forward to see more publications in this.
All right, thank you, Dr. Su.
Yeah.
So due to time constraint, we'll end the Q&A session over here. So for those questions that we didn't get to answer just now, we will email the answers back to you after this webinar. So thank you once again, Dr. Su, for the great presentation, and to all of you who joined us today. And I hope you find this session today beneficial to your work. So thank you, everyone.